want to know, is that homozygous dominant or is that heterozygote? What do you think I would mate it with? If I mated it with another heterozygote, would that give me an opportunity of seeing? Yeah?
But then if it's true homozygous, that means the dominant genes to show all the time. So
Okay. So if it's homozygous dominant, and I mated it with the heterozygote, I'm going to get my Punnett square will give me.
No, you want the recessive because then you see the 50%.
Exactly, because this is going to give me all dominance. You're absolutely right. Okay? But if it was a heterozygote condition here, then I would get a 1/4 chance of getting that recessive out. But if I mated it with the recessive, I still would get either all dominant alleles out, or I would get, if it was a heterozygote, I would get 50-50 coming out. Okay? So you really want to do a recessive allele to determine what that unknown genotype is. Okay? To figure that out. This is what we call a test cross. It's basically, if you use the heterozygote, you don't even know, first of all, if you're pulling out a heterozygote plant to use it with. I don't know if I'm heterozygote or homozygous dominant for my earlobes. Okay? But to figure out what my genotype is for my earlobes, I could mate with somebody that has attached earlobes. And if I have 5 kids and one of them showed up with detached earlobes, well, then I know I'm heterozygote. If they all showed up with attached earlobe or detached earlobes, then I don't know. I still don't really know what my genotype is, but most likely it's dominant versus recessive. We have been talking about Mendel and his work with the garden pea, how his work was very significant at the time, but nobody appreciated it, nobody accepted it. He did his monohybrid crosses and his dihybrid crosses, and after performing those experiments, he came up with 2 laws. The law of segregation, basically stating that chromosomes or these traits will separate during the formation of gametes, and also the law of independent assortment, where he said that these traits, as they separate, will do so independently of each other. They will not depend one trait on the other of where they go in the gametes. They will separate independently of each other. And his work truly was not understood until there was an understanding of meiosis. And that didn't happen for about 40 or 50 years later. He was in the middle of the 1800s. It wasn't until the turn of the 1900s that meiosis was really, truly understood. And then somehow, somewhere along the line, there was a scientist doing some kind of research, was in the library, came across Mendel's work, came across these experiments with the garden pea that he had developed. And he goes, wow, this is kind of stuff that I've been working on. This is what genetics is all about. This explains, or meiosis explains, that what he was doing actually is true. But Mendel got very lucky, like I said, in choosing that garden pea. Because even after his work was discovered, there were some exceptions to his work that came out. They were like, okay, well, complete dominance happens. This type of inheritance, you have the trait, you don't have the trait. That recessive allele can be hidden, can be completely overshadowed by this dominant allele. And that does happen with a lot of traits. The clasping of your hands, the widow's peak, the rolling of your tongue. All of those are complete dominance. But there are cases, and had Mendel chosen something else to work with, he may not have come up with these results. But there are cases where blending actually does occur. And there are cases where those recessive alleles are not hidden. Okay? So what we are doing here, Mendel basically, with his complete dominance or simple dominance, kind of set the bar, kind of set the standard. But there are variations. It's science. There's always exceptions to everything when it comes to science. On one side, we have what's called incomplete dominance. Now, incomplete dominance is a process or is a form of inheritance where there is blending. There is blending that actually happens when we talk incomplete dominance. An example of where you can see incomplete dominance is in snapdragons. Anybody know what snapdragons look like? They're real pretty flowers. They're like these long flowers. They got little pops like they do in floral places. I don't know if they naturally grow or if they can grow them in, you know, the nurseries or anything like that. But I know you could get them in floral arrangements and stuff. But snapdragons are really pretty. The dominant allele in a snapdragon is going to give you a red color. For example, we'll use red. Okay? And the recessive trait in snapdragons is white. And had Mendel picked up and was a botanist that liked growing floral arrangements versus, you know, garden peas, he may have chosen the snapdragon. And if so, then of course this blending theory would have come out. Because when you mate two snapdragons together, a red one and a white one, for example, if this is our parent cross, that first generation is always going to be what? If this is, you know, if this is dominant and this is recessive, what's that first generation always going to be? Genotype. Heterozygote, right? Well, we would assume, based on Mendel's theories in his work, that this first generation would be red, right? If complete dominance was occurring. But we're talking incomplete dominance, and in incomplete dominance, blending occurs. So what color do you think this first generation would be? Pink. These flowers turned out to be pink. And in that situation, then you know exactly what the genotype is for all of the different flower colors. Red is dominant, homozygous dominant. White is homozygous recessive. And pink then is going to be heterozygote. So that second generation, what do you think you're going to come up with? If we were coming up with a 3-to-1 ratio with complete dominance, you're going to come up with one red. You're going to actually come up with two that are pink because they're heterozygote. And you're going to come up with one that is white. Okay? So this is something that is an example of blending. Now, would you ever have true breeding plants that were pink? No. You couldn't do that, could you? Because that heterozygote condition is not a true breeding condition. We can't mate. If we mated two pink snapdragons together like we have here, we're going to come up with some red, and we're going to come up with some white, and we're going to come up with some pink too. So in incomplete dominance, you are actually seeing the heterozygote condition, and you can say, I'm heterozygote. I know what my genotype is. Normally, the only time you can say that is when you're recessive, right? When you have attached earlobes, when you can't roll your tongue, when you have a straight hairline. Those are all recessive traits. You know exactly what your genotype is. But those dominant traits, you don't know. Am I homozygous dominant or am I heterozygote? I don't know. I have to go forward. I have to go back generations to figure it out. To try and figure it out at least. Okay? Here you can do it. I'll give you an example that's more realistic probably than snapdragons. Curly hair and straight hair. Okay? There's a difference, right? There's curly hair and there's straight hair, and then in between you have wavy hair. Curly hair is dominant to straight hair. When you mate somebody that has curly hair with somebody that has straight hair, all of the children will have wavy hair. And now I'm not talking coarseness. I'm not talking, you know, like the thickness of the hair. I'm just talking about, is there a wave or is there a curl? Or is it just straight? Okay? My family is a great example. Once again, I love family history. Because my parents both have wavy hair. My mom doesn't have real curly hair. My dad doesn't have real curly hair. They both have a nice wave to their hair, though. Now, that means they're both heterozygote, right, in their cross, which means they're going to have one child, or the percentages, one will have curly hair, two will come out with wavy hair, right, and one will come out with that straight hair. That's exactly what happened in my family. There's 5 of us. My oldest brother, the hair that he has left, which isn't much, but when he had it, was poker straight as straight can possibly be. My youngest brother, he's got the curls that he tries to flatten out as much as he possibly can, okay? And me, my sister, and my other brother have pretty much wavy hair. I've got more curl probably than anybody else, but we're all pretty much in this wavy hair situation. And it's amazing how genetics plays itself out. But if you look at your own family history, if you got somebody that has curly hair and somebody with straight hair, and you could probably see this kind of breakdown. So the odds of us getting the wavy hair, there was, out of 5 kids, we had this Punnett square down to a T, and then that 5th one came along and just added into the 50% chance. You've got a quarter chance of one or the other, and then 50% of the wavy, and that's how it fell out. Okay? So large families work out well when you're trying to do pedigrees and things like that. You can see all these differences. But that's incomplete dominance. Incomplete dominance is a blending. Curly and straight makes wavy. That makes sense. Red and white makes pink. That makes sense. That's incomplete dominance. The key is this dominant or this recessive allele is not hidden. It still gets to express itself a little bit. This dominant allele is not overpowering. It's not that almighty dominant trait saying, forget you, you disappear, like in complete dominance. It actually shows itself. So you can actually see all 3 genotypes in the offsprings. Any questions on that? Well, that's one side of the spectrum. If we come to this side of complete dominance, we have what we call codominance. If we come this way, codominance occurs. Now, what codominance means is that there is no true dominant allele. Both alleles represented are shown as dominant traits. One of the best examples of this is a multiple allele known as our ABO blood typing. Okay? ABO blood typing. Anybody know what their blood type is in here? What's your blood type? A positive? O positive? AB. Excellent. B positive? O negative. AB also? Okay? So, what that means is we've got an A, we've got an O, we've got ABs, we've got a B back there. The rest of you, try and find out your blood type. It's an important thing to know. Okay? Everybody should know their blood typing, especially Sarah, knowing that she's O negative. You know the complications behind that, I'm sure. And we'll talk about that in a second. Well, codominance with our ABO blood typing is very important because what it does is our blood cells contain proteins. They're known as immunoglobulins. And on these immunoglobulins, we have these markers that detect what our blood typing is. Okay? If you have type A blood, you have 2 variations of that type A blood you can be. You can be dominant for type A blood, and I'm putting it on these eyes because it's represented as the immunoglobulin. Okay? You could write it out like, AA if you wanted to. But it's the markers, these alleles, are actually found on these proteins, on these immunoglobulins. So that's how you usually see it written. So you could be homozygous dominant for type A, have 2 A alleles on your genes. Or you may have 1 A allele and be recessive. Okay, which, you can write this as like maybe AO. So here you're heterozygote. I said recessive, didn't I? Heterozygote or homozygous dominant for type A blood. So Jenna could be heterozygote or homozygous dominant. If you have type B blood, it's the same thing. You could be dominant for both alleles, or you can be heterozygate for both alleles. If you have type O blood, you are actually recessive for both alleles. You carry no markers on your blood. You will not react form antigens and antibodies with those markers because you don't have any. Okay? Now, there's 2 of you in here that actually have type AB blood. And what that means is you actually have one allele that is type A, and you have one allele that is type B. Both of these are dominant alleles, aren't they, to that O allele, or that recessive. But when they are together, they are both expressed completely. They both have complete dominance. A does not overshadow B. B does not overshadow A. So when you have type AB blood, you are carrying both alleles. You have both alleles on your immunoglobulins, showing this co-dominance. That's what it means. It's sharing that dominant trait. Okay? Any questions on that so far? When they used to do blood testing, you know, years ago before you got married and stuff like that, they actually, you know, wanted to make sure you weren't marrying your brother, basically. And they also wanted to make sure that you weren't in a situation like Sarah. Now, AB, O positive, A positive, you said? O negative. Well, that positive or negative factor associated with our blood typing, that's complete dominance. Positive is dominant to negative. It's found on another protein, on what we call the RH factor. Okay? And the RH factor, you're either positive for it, where you have 2 positive alleles, or you're heterozygate, where I should just be using, instead of doing it like that, let's just do it like this. You're either positive, showing 2 positive alleles. Okay, I could put those up there. Or you're heterozygate, showing 1 positive and 1 negative allele. Or you have 2 negative alleles, which is the homozygous recessive condition. This is complete dominance. The problem that arises if you are a female and if you have negative type blood is when you become pregnant. If you're a female with negative type blood and you become pregnant with a child, a fetus, that has positive type blood, your body sees it as being foreign. And it will reject it, meaning it will abort that baby, have a miscarriage. If it's your first child, your body hasn't built up the antibodies to it yet. And so what they do, blood testings before you're married, was testing to see if you were positive or negative. If you were negative, they would make you aware of this situation. Since the 50s, that's what they've done. Okay? If you are negative and you're marrying somebody that has positive type blood, now, it doesn't matter if the male is negative, it's the mother, the female that's negative. And she becomes pregnant. That first child, if it's positive, whether it's positive or not, it doesn't matter. About 6 months into the pregnancy, that mother will receive a shot known as a RhoGAM shot, okay, which will help her immune system not become activated to this positive type baby. Now, there's no mixing of the fetal and the maternal blood until delivery. There's no mixing of that blood until delivery. And after delivery, the mother receives another shot of RhoGAM to say, okay, you're not going to produce antibodies, because if she was pregnant with a second child that was positive type and did not have the shot, she would abort it. She would miscarry it. And so you need to be very consciously aware, and that's why I say it's pretty important to know your blood type, especially if you're a female, when it comes down to it. You never know if a trauma occurs, but also, you know, when you're trying to get pregnant. That's one of the first things they do at your prenatal checkup, that very first prenatal checkup, is check your blood typing. And if you happen to not know your blood type, given a scenario, you don't know your blood type, you become pregnant, you have a miscarriage, you don't tell the doctor. You're negative type, you have a miscarriage with a positive type baby. You could end up not being able to have children in the future if you're negative and that baby was positive because you didn't inform the doctor about that baby or whatever. You get where I'm going with that? So you need to be careful. You need to be consciously aware that there can be complications later on down the road. Okay? But this is positive and negative is complete dominance versus our ABO blood typing, which is codominance. Now, AB blood type is the rarest blood type. You're not AB. You were AB. AB blood typing is our rarest blood type, but it's known as the universal acceptor because it has both markers. It can accept all three blood types without any problem. So if you guys are ever in an accident and you need a major blood transfusion, whatever blood they got, you're good. You're good to go. Okay? Type B blood can only accept type B blood, type O blood. Type A blood can only accept type A or type O. And us lowly O type people, we can only accept type O blood. We could give it to everybody, though, exactly. We are the universal donors. Type O blood is the universal donors, and O negative is the cream of the crop, you know, because everybody under the sun can take O negative blood. And whenever you are in this serious accident and they have no idea what your blood type is, that's exactly what they do. Get me some O neg. You see the drama shows, you know. Get me some O neg stat, right? Because that O negative blood is not going to react with anybody. And once they determine, well, she's type B blood, she's B positive or whatever, then they can start pumping in the B positive. But it's that O negative that's the cream. But O is the universal donor, AB is the universal acceptor. One of the greatest paternity tests or paternity trials was based on these ABO blood typing. Back in the 20s, I think it was, or something like that, Groucho Marx was being sued for paternity. And he had type A blood, and the mother who was suing him had type O blood. And when it was brought to court, it was found out that the baby had type B blood. What do you think happened? It got thrown out. How come? Because type O and type A blood could not give you a type B child. And so it was thrown out saying that, well, he's not the father of that child. There's no way he can be. If he has type A blood and that child is type B, he cannot be the father when the mother was type O. Now, what if the child was type O? Could he have been considered the father? Yes, he could have. Because even though he was type A and the mother was type O, he could have been heterozygous. Meaning he passed on this small I, and mom passed on the small I because that's all she had, and they got a child that had type O blood. So he would have been paying for this child his entire life had he been type O. If you have somebody that is type A and somebody that is type B, you can actually have an offspring with all four blood types because they can be heterozygous for both conditions, and in turn you end up with a child that has type AB blood, a child that is type B, a child that is type A, and a child that is O. Why? Complete dominance is going to give you this. And so your parents had to be type A and type B to give you guys type AB blood. But if you have any brothers or sisters, thing is, are they all type A or AB, or do you have one of each? Or, you know, if this was a dominant allele here, then you would have two ABs and two A's or something like that, okay? So this blood typing gives you many variations when it's something that's codominant. These are multiple alleles, okay? These are considered multiple alleles. Multiple alleles being expressed at the same time. Any questions on that? Okay, well, that's easy enough. The rest of, well, not the rest of this chapter, but some of this chapter talking about pleiotropy and epistasis and things like that, I'm not going to go into. But I do want to talk about some patterns of inheritance. We talked about our chromosomes, how we have 23 different types of chromosomes. 22 of those chromosomes are known as autosomes, and the other pair is known as our sex chromosomes. Well, the way we inherit our different traits is based on what chromosome they come from. Now, it doesn't really matter one way or another if you can roll your tongue. It has no significant difference in your life, period. But these genetic tests were set up or to determine, go through like what Mendel had discovered. These things were developed for genetic testing to determine what your percentages were of having a child with this disorder or having, the chance of having this disorder yourself or something like that. Okay? That's what genetic testing was developed for. And there are genetic counselors that continuously will meet with patients saying, okay, well, here's the odds. This is the percentage of you having a child with this certain disease. And what they do is they look at family history. If you've ever done a family tree before, you know how it branches. Like it starts with great-grandparents and what kind of kids they had. Say there was 4 kids on each side, and then they married and they had kids, and then they married and they had you. Okay? And you develop this family tree or this family history. Well, that's what genetic counselors do too. They'd establish this chain of genetic characteristics. They do research into your background saying, well, do you know what your parents' conditions were here, or maybe even test the parents. Or now, even with modern technology, they do your own karyotype. And they test your karyotype and determine what your allelic combination is. Are you homozygous dominant? Are you heterozygote? Or are you homozygous recessive? So they will go through these tests to determine your odds or your chances. Usually, there's a family history that is stemming this information. Okay, now I shared with you that I had several miscarriages before. There's no extreme family history of these kinds of miscarriages. So that they didn't know where it was stemming from. That's why they did the chromosome test. They wanted to look, is there anything abnormal in my family or in my husband's family that could have caused these miscarriages to occur? Thank goodness there was nothing. Okay? But there are a lot of patients that there are histories like that. There are problems like that. And so what they establish is a pedigree. They establish this flow of information. This, for example, is, well, actually, let me see. I don't have the one chart that I want. When you're establishing a pedigree, what you usually do is you show the males being represented as squares, okay, and the females being represented as circles. And then you code them in a certain way. Like, these are coded differently. This here, they were looking for the dominant allele. So if you carried that dominant trait, they colored in the box, saying you were affected by it. You had that dominant trait. This is for widow's peak. Here's 2 families. The fathers had widow's peak here, and the mother had a straight hairline. And here the father had a straight hairline, and the mother had the widow's peak. They had their children. This family had 4. This family had 2. Well, those 2 children met. They married, and they had 2 of their own children. One of them had a widow's peak, and one of them had a straight hairline. We don't know for sure if that one with the widow's peak was dominant or heterozygote for the widow's peak, because she didn't have her children to look at. Because we see what these children are here, we can go back and determine that, well, this was a heterozygote condition here, because they had a child that was homozygous recessive, just like my son clasping his hands with the recessive way. So you were able to go back a generation to determine what your or forward a generation to determine what your genetic history was all about. So the colored in box or the colored in circle shows that that person is affected, shows that trait. Here, on the other hand, they're using the recessive as showing the trait. If you are recessive, then you have a colored in box being affected. And you can see here for attached earlobes is that recessive characteristic. This family was heterozygote. They had 4 kids. 2 of them showed that recessive trait. But we said before, it's a 25% chance every offspring. Not just because I had 1 with attached earlobes doesn't mean that I won't have any more with attached earlobes. Okay? I still had 2 of them with attached earlobes and 2 with detached earlobes. Once again, you can see here, they don't know what this person's genotype is for that dominant allele because there's no offspring. But this one they can determine they're heterozygote because the offspring has an attached earlobe. Okay? So this family history, looking at your family, going back, and if it was the regular semester, sometimes I send you home over Thanksgiving and say, okay, look at your family history at a family characteristic and come back with a pedigree. But we don't have time to do that. I don't have a nice break to do that over. But it's a neat thing to do. Pick a trait and say, okay, did my parents have this? Did my grandparents have this? Look at pictures or whatever, or, you know, if they're not still alive. Do my children possess this? Do my grandchildren? None of you have grandchildren. But, you know, do I have a pedigree that I can follow? This form of inheritance here, the earlobes or the widow's peak, this is known as an autosomal recessive for autosomal recessive for earlobes because that's what's being expressed, or autosomal dominant for the widow's peak, dominant for the widow's peak form of inheritance. Okay? Now, like I said, it makes no difference in your life. You're not going to become a movie star because you have a widow's peak, or you're going to be turned down because you have a straight hairline. Okay? It makes no significant difference in your life about the widow's peak. But it does make a difference when you're talking about a characteristic like cystic fibrosis. Cystic fibrosis is an autosomal recessive form of inheritance. Now, this isn't in your book, but you pretty much have an idea of what's going on here. An autosomal recessive type of inheritance means that both parents have got to be carriers. They both have to be heterozygote. A carrier does not express the gene, does not express the trait, because that dominant allele completely overshadows it. And by that dominant allele overshadowing it, you don't know it's there unless there's a family history. But 2 heterozygote parents, 2 carrier parents, have a 25% chance of having a child with that affected allele, meaning 2 recessive alleles. Okay? So what that means is if there's a risk of having a child with cystic fibrosis, 75% chance that you'll have a normal child. But there's that 25% chance that you'll have a child with cystic fibrosis, which is a disease that is pretty severe. It's a disease of the lungs where you literally, with these children, right, I mean, you're like, they build up mucus. You got to smack them on their backs really hard to try and break it up, get it out, things like that. They have a lot of medications now that really help these children. Years ago, they only lived maybe into their teenage years. Now, they're into their 30s and 40s and things like that with medication, which is great. I had an AP student last semester. Her best friend has 2 daughters, both of them with cystic fibrosis. Both of them with cystic fibrosis. So, they, you know, it wasn't like they planned on, the second child was kind of an, I don't want to say an accident, but it wasn't something planned because they had one child already with this disease. Now, the sad thing about them, they're both like very young, 14 and 12 or something like that. But they have contacted a a virus or something like that, something that is basically irreversible. They're not allowed to have any contact with any other cystic fibrosis patients because this disease, they call it almost like the typhoid Mary of cystic fibrosis, meaning that's it. There's only a matter of months to a few years before they're going to both pass on because of this extent of where it's gone, something like that. I don't know. But just because you had one child doesn't mean you won't have a second one or even a third one or a fourth one or something like that. I had a student a couple years ago whose father had two sisters that died of cystic fibrosis. He was genetically tested to be a carrier. She was genetically tested and was normal, which is a good thing. She wasn't even a carrier. She didn't have to worry about passing it on. But both of her aunts had died of cystic fibrosis at a very young age. So it's when they do genetic testing, that's what they're looking at. Tay-Sachs disease is an autosomal recessive form of inheritance. Tay-Sachs affects, more often the Jewish population or the Jewish nationality than other nationalities. But it is from intermarriages and things like that starting to spread more. Okay? But it is an autosomal recessive form of inheritance. There are carriers out there that are passing it on. Sickle cell anemia. Sickle cell anemia is an autosomal recessive form of inheritance. If you are a carrier for sickle cell, it means you have the sickle cell trait. You have pretty much a normal lifestyle because a sickle cell anemia creates red blood cells that are not spherical in shape, but more sickle-celled in shape. And what happens is these sickle cells get clogged up into the red blood cell or the arteries and veins and stuff. They kind of stick, and it blocks the room for our normal red blood cells to go through. So if you have the sickle cell trait, being a carrier, you can pretty much function well. You might tire easier because you don't have as much red blood cells to carry the oxygen. Or your arteries and veins may get clogged more by the sickle cell, and so you have to try and wait until they unclog and things like that. But you can pretty much live a normal life. However, if you have both recessive alleles, it can sometimes be fatal because now you're not carrying any oxygen at all because you've got all of your cells are sickle-celled in shape. Okay? So it's usually fatal. Sickle cell anemia is a disease that was more prominent in the African American population because it developed as a defense mechanism basically against malaria. If you are sickle cell, you will not get malaria. It was Northern Africa, a tribe up in Northern Africa. The population was dying out from malaria, but there were some that were surviving. Some that didn't die passed on this sickle trait. And in turn, it's like an immune response or a natural selection to malaria. So if you're exposed to malaria, you're okay. If you're not, then, But once again, sickle cell anemia is a characteristic that is getting into other populations, other nationalities, Caucasians, Hispanic, or whatever, from intermarriages, okay? Starting to see it more dominantly or more frequently. Okay? So that's a recessive form of inheritance. There's also the autosomal dominant form of inheritance, where the normal condition is actually the recessive. And the affected condition is either heterozygote or homozygous dominant. If you have an autosomal dominant form of inheritance, basically 50% of the offspring can be affected by that type of inheritance. One of the types of autosomal dominant forms of inheritance is one that strikes very late in life. And well, I shouldn't say very late in life, but later on in life, in the 40s. in the 50s, and it's known as Huntington's disease. Huntington's disease is a debilitating disease. It basically, your neuromuscular system literally shuts down. It starts off as shakes, you know, where you're dropping things and stuff like that, kind of similar to Parkinson's, but yet, your muscles just start to get weaker and weaker and things like that. Michael J. Fox has Parkinson's, okay? But Huntington's disease is an autosomal dominant form of inheritance. Now, by the time somebody's 40, 45 years old, what have they usually already done? Been married and had kids. So there's a 50-50 chance of passing that allele on to your offspring. I have a student, or I had a student several years ago, and I actually saw him the other day because he works at the bank up the road. But his grandmother has Huntington's disease. His mother and his aunt had never been tested to see if they were carriers or if they were affected basically by it. And his response was he didn't want to be tested either. He did not want to know if he was going to die at the age of 45, 50, something like that. He didn't want to know that. If he has it, he has it. At the time, his mother had not shown any signs of having it, but you never know, okay, somewhere down the road. So it's a personal choice and a personal decision. Do you want to know if you are affected with this autosomal dominant form of inheritance? Did you get it? It's a choice you have to make deep inside, right? If you don't find out, do you want to risk having children of your own? It's a choice you have to make deep inside of yourself. Okay? So just giving you an idea of some of the things out there. How widow's peak does not affect your life, but a disease like Huntington's, where you may not even know. Maybe you didn't know your grandmother had it. And now your father is showing signs of it. You've already had 5 kids. You know, or your daughter's ready to walk down the aisle or something like that. Do you tell them? You're just diagnosed with it. Do you tell your daughter that's ready to go down the aisle and wants to have kids right away? You know. But sometimes people died in the wars or something like that. You never knew it was passed on. Who knows? Okay? So it's just something that you want to keep in mind. Great food for thought, isn't it? All right, there's one more type of inheritance that I want to talk about. We talked about inheritance on just our 1 through 22 chromosomes. Any of those chromosomes, we can go through these inherited forms of diseases. But there's one other one that I want to talk about, which are sex-linked inheritance. Now, if a gene is linked to the sex chromosome, it can either be to the X chromosome or to the Y chromosome. All that we're going to look at are X-linked recessive forms of inheritance. Okay? Just some X-linked recessive. Doesn't mean that it can't be X-linked dominant, or it could be Y-linked recessive or dominant. But we're going to just look at a few X-linked recessive forms of inheritance. When genes are linked together in general, that means they're going to show up together all the time that way. And so when it's linked to the X chromosome, that means anywhere there's an X chromosome, which is basically in males and females, you're going to see this form of inheritance. But when it's X-linked recessive, it usually shows up more predominantly in the male population. And the reason being is that males only have 1 X chromosome, whereas females have 2 X chromosomes. And so that 1 X chromosome is going to show that recessive allele. And whereas the female is going to show that dominant allele on the other X chromosome quite often. So when different things that are X-linked recessive, male pattern baldness, that's that I'm losing my hair up here, I'm keeping the band around here type of thing. That very typical, not just losing some hair back here, but that very typical male pattern baldness of going, receding hairlines, eventually leading to nothing. Once again, my brother who used to have poker straight hair, male pattern baldness. He loves it when I talk about him too, I'm sure. Another form is hemophilia, which is a blood clotting disease, very prominent in the royal family over in Britain. Actually starting to breed out because in the royal bloodline, you can only marry and mate with royal bloodline. Okay, you have to be royal in that bloodline in order to mate. And so it was very prominent a couple centuries ago, very much out there, showing up in a lot of the males, hemophilia. But it's starting, the royal family that's in office now, it's pretty much out of their bloodline. Okay. Red-green color blindness. Red-green color blindness, another form of X-linked recessive. So what these are doing, what these are showing is that reasons why we see it in the males more often. If you have a female, let's use red-green color blindness. No, it doesn't go along with the bar body because it's affected during development. It actually affects the proteins during development type of thing.
So if this was on like the X that was turned off, would you still show these traits?
It would still show these traits, yes. Okay. So if you have a female that has normal colored vision, okay, normal color vision, and she mates with a male that is colorblind, okay? She mates with a male that's colorblind. So if we did the Punnett square, here she's normal. All of their children are going to be normal, right? These two daughters here, they're going to end up now being carriers, but they're still going to have normal color vision. They're still going to see normally. And these males are not affected at all. Okay? So now, here's your mom. Okay? This is your mom. She has normal color vision, but she's a carrier. She's a carrier that mates with your dad who has normal colored vision. Okay? So, let's see what happens. The daughters, one of them will have normal vision. The other one will have normal vision, but now she's a carrier. Still normal vision. One of the sons, normal vision, and here you are, colorblind. You see that? So 50% of the sons are going to be colorblind, aren't they? When you're working with X-linked characteristics, you have got to put them on the sex chromosomes. If I just did RR and, you know, like, if I just did a cross like this, or, you know, like this or whatever, I would not come up with the same answer, would I? Okay? So you have to link it to the X chromosome. Put it and do your Punnett square with the X chromosome. Male pattern baldness. You're going to be bald. You're going to have a normal thick head of hair. If you go back, it goes male, male, male. To your mother's father. There you go.
Because my cousin's like, I see him at the barbershop all the time because we were locally family the same way. He's losing all his hair. He's been losing since he was probably 18.
Yep. That's how my brother was. The one with the straight hair. By the time he was 22, it was like going.
He lost his, and then my mom and my aunt, so I guess they're carriers.
They're carriers. He got it and you didn't, exactly. So if they were carriers, maybe your mom's not a carrier. Well, no, yeah, because you said your grandfather had it, right? So you, both of your aunt and your mom are carriers. You got lucky. So far. Huh? The grandfather and your dad's father?
Yeah. And no, I guess we'll just have to say my mom
You look at your mother's father.
and my dad's father.
Your dad being bald won't affect you because your dad gave you your Y chromosome. So whether he was bald or not, you're normal. Okay? Now, if your mother's father was also bald, that's where you might have a risk. Now, females are not immune to this. So say your mother is a carrier because your grandfather was bald. Okay? So your mother is a carrier and your father is bald. Right? So now what you've got is a sister that's going to be affected. Oh, wait, no, that's a small b. You've got a sister. This one is a carrier. This one now is going to be affected. This one, you could be normal, or you can be affected. You got a 50-50 chance. Keep wearing those hats. Huh? Oh, you're just making an example. But females can show red-green color blindness, hemophilia, male pattern baldness. Now, when a female shows male pattern baldness, they don't normally lose all of it. They will get a deep receding hairline. They will get real thinning hair on top, but they won't show that, banding pattern. Okay? Now, in my family, my grandmother's brothers, boom, it's like there was 11 kids, 7 of them were males, and every one of them, I couldn't, is that Uncle Dom, or is that Uncle Paul, or is that Uncle Nick? Who, because they looked all the same. When you're 10 years old and you're going to family reunions, you're like, which one, who are you, So my grandmother actually passed on that trait to my mom, and my mom passed it on to my brother. And he's bald. He shaves. It's what most men do anymore, right? Michael Jordan look. It's a nice, smooth look. I like that. Huh? Yeah, and you only have half of it to shave anyway, right? But he still has a little bit up on top here, but he started at like age 20, 21 with this major receding hairline. He had the straight hair to start with, and it just went downhill from there. Now my middle brother, he's starting to get this deep hairline here, but he hasn't lost the rest of it. And the little one with the curls, ain't ever going to happen. Okay. Any questions on these patterns of inheritance? Autosomal recessive, autosomal dominant, X-linked. You got to make sure when you're doing X-linked forms of inheritance, you put them on the sex chromosomes. That's the important part, or you'll get totally screwed up on them.